The family of tyrosine protein kinases that is the receptor for extracellular ligand epidermal growth factor, or EGF, has 4 members, erbB1 through erbB4. The best known of these is erbB1, also called EGFR, which is the major receptor for EGF in many normal cells as well as in tumor cells. The physiological activation of an EGF receptor and the transmission of the signal into the cell is a process of several steps. After binding the ligand, the receptor monomers dimerize. The intrinsic tyrosine kinase is activated, and several tyrosine residues of the intracellular domain are phosphorylated. Phosphorylated receptor tyrosine protein kinases are able to recruit cytoplasmic proteins, which activate intracellular pathways and thus transduce the signal into the cell. Pathways activated by receptor tyrosine protein kinases include the PI3K ACT pathway and the MAPK pathway. Via these and other pathways, the EGFR system activates proliferation, growth, differentiation, migration, and cell survival. There are several different ways that EGF receptors can be activated in tumor cells. In many breast tumors, the erbB2 gene is amplified or overexpressed. The amplification or dysregulation of the EGF receptor gene results in the increased density of the receptors in the plasma membrane. The receptors dimerize spontaneously, independently of ligand binding, leading to inappropriate signaling. Point mutations have been found in the transmembrane domain of a very small number of tumors. These mutations may result in the spontaneous dimerization of receptor monomers, independently of ligand binding, leading to inappropriate signaling. Mutations in the cytoplasmic kinase domain of the erbB1 gene are found in lung tumors. Such mutations may lead to the constitutive activation of the kinase and to inappropriate signaling. As previously mentioned, EGF receptors must dimerize to become active. However, they do not have to form homodimers. erbB2, also called HER2-Neu, is not activated by an extracellular ligand, but by heterodimerization with a monomer of a different EGF receptor, for example, erbB1.